Hello, my name is Henry Lee, and this poster is about designing a benchtop-focused ultrasound system for drug screening investigations in central nervous system. So, BBB opening with focused ultrasound and microbubble is a promising drug delivery strategy for treatment of neurological diseases. An MRI-guided FUS system has been the standard for this procedure due to its high image quality, but it's limited due to its high cost and lack of portability. So a ultrasound-guided FUS system can be an alternative in order to address the limitations of current MRI-guided FUS system. And the goal was to design a cost-effective, portable, and closed-loop system for BBB opening with microbubble enhanced focused ultrasound, all of those with quasi-MRI targeting accuracy. The schematic of the whole system is shown on the left, and the system is composed of two subunits, imaging unit and therapy unit. In imaging mode, the system first raster scans around the mouse head with 3.5 MHz central frequency imaging probe. The recorded echoes are then analyzed to obtain a 2D contour of the mouse head. From the 2D contour, the location of the eyes are obtained using the eye locator placed on the mouse beforehand, as shown in the right figure. Then, a target coordinate, say tumor location, with respect to eyes is determined using a contrast-enhanced MR image. Finally, the system guides therapeutic FUS transducer to the target location given the coordinates and performs the therapy. The targeting accuracy was obtained to be less than 500 micrometers. To implement our recently proposed closed-loop controller for BBB disruption, we needed to train the controller. To do so, we first hypothesized that harmonic level and bubble echo signals obtained from passive cavitation detection is correlated with microbubble oscillation amplitude. Reyleigh-Plesset equation is an ordinary differential equation that governs the dynamics of a spherical bubble in an infinite body of incompressible fluid. In this model, with some assumptions, we validated our hypothesis by observing the fact that increasing oscillation amplitude is followed by increase in level of harmonic components in frequency spectrum of the bubble oscillation. Secondly, we hypothesized that the harmonic level is correlated with number of microbubble oscillating. To test this hypothesis, we observed the harmonic levels during in vivo BBB disruption experiments after a bolus microbubble injection. The harmonic level, third harmonic level in our case, showed tendency to decay after a bolus microbubble injection during sonication with a constant pressure. This suggests that as the number of microbubble decays as the mouse heals from microbubble, the corresponding harmonic level also decays. Thirdly, we hypothesized that the harmonic levels during BBB disruption is correlated with the BBB permeability. To test this hypothesis, we dedicated 5 mice with varying input focal pressure, where each of the mice were treated at 4 different locations on the brain with the same pressure. Here, to quantize the BBB permeability, we'd like to introduce K-trends, which is a measure of capillary permeability obtained using dynamic contrast enhanced MR perfusion. We found out that the mean of the harmonic levels for 40 seconds after microbubble arrival to the brain was positively correlated to the corresponding K-trans value for that target location, which suggests that we can estimate the BBB permeability for a given harmonic level. Fourthly, we hypothesized that the harmonic levels during BBB disruption can be controlled. To test this hypothesis, we used our recently proposed harmonic level controller algorithm. Essentially, the controller takes in a model that represents the tendency of the harmonic levels with respect to increasing pressure, which is then used to calculate the pressure increment to a target level based on the model. Figure on the left shows the model that is obtained from in vitro model from the paper, and the figure on the right shows the model that we have obtained by sweeping pressure on the mouse brain. And from our previous data, we set 2 target levels, one level from where only third harmonic was present, and the other from a region where some second ultraharmonic was present. The controller algorithm was applied for 40 seconds after microbubble arrival to the brain, and we were able to maintain the target levels for each of the different target levels. And our final question is that since the BBB permeability is correlated to the harmonic level, and that the harmonic level can be controlled, can we control BBB permeability by controlling harmonic levels? This hypothesis is currently being investigated and will be our next step. And here is a brief summary of our lab members and acknowledgements. Thank you for your time.